call him.
It's okay. It's your.
Okay. Thank you.
Let's wait for 1 minute. Okay, good afternoon. Let's get started. Okay, so welcome. It's my great pleasure to welcome you to the doctoral defense of seminar of MD Khairul Islam. Khairul received his bachelor degree in information and communication technology from Islamic University, Bangladesh. In 2020, he joined my group in the fall 2022. Over the past 3 years, he has been working at the intersection of biology, computational science, and artificial intelligence. During his PhD study, Khairul has demonstrated exceptional productivities and academic excellence. He has authored a total of 21 publications, including 13 peer-reviewed journal articles, 6 conference papers, and 2 book chapters. He also has 1 patent. In addition, he has delivered 2 invited talks at the international conference, both of which was very well received. Khairul has maintained an outstanding GPA and was honored with the Dean's Award in 2026. He has also secured a postdoc position at Outreach National Lab, DOE, to work on the AI modeling. Today, he will present his dissertation titled Computational and AI Framework for Identifying Key Regulatory Genes and their targeted genes in plants and humans. So please join me. Welcome, Khairul.
Thank you. Good afternoon, everyone. I hope I am audible enough. How about Zoom? So first of all, I would like to thank everyone for being here today, especially Professor Hairong Wei and my committee, advisory committee member. And my name is Mohammad Khairul Islam, and I'm originally from Bangladesh. and over here I'm pursuing PhD in Computational Science and Engineering under the supervision of Professor Hairong Wei. And today I will show my PhD defense seminar, and the title is Computational and AI Framework for Identifying Key Regulatory Genes and Their Target Genes in Plants and Humans. My research focuses on a very simple question but very important one. Given thousands of genes in a genome, what are the genes that are most important, that really matters? The one that drive disease, control development, and identify downstream gene of a signaling pathways. In last few years, I have tried to build several computational framework to tackle those questions, and today I will walk you through all 3 chapters. And let's start with a bird's eye view of all chapters. So chapter 1, DIGAF, focuses on identifying biomarker prioritization using transformer model. Chapter 2 focuses on identifying downstream gene of a signaling pathway using pseudo-peak analysis. And chapter 3 focuses on identifying transcription factor in regeneration using network biology. Chapter 1. The title is Dynamic Gene Attention Focus, DIGAF in short, enhancing biomarker identification through dual model attention network. This work actually funded by Michigan Department of Health and Human Services, and this was published last year in Bioinformatics and Biology Insight. So let's start with the question, why biomarker gene really matters? So when we talk about a molecular biomarker gene, we actually aiming for multiple real issues, such as early diagnosis, especially when symptoms are overlapping or very similar. Second, guidance treatment, right treatment, therapeutic treatment for the patients. Third, prognosis, predicting the risk and risks of the diseases and progression. And fourth, monitoring, tracking diseases activity and treatment response over time. So the challenge is not just to identify statistically significant gene, but also we want to identify biomarkers that are reliable and useful for decision making. So now the question is, how do people actually identify biomarker genes in practice today? So traditionally there are 2 broad directions to do that. On the experimental side, we use high throughput sequencing data like RNA-seq, single cell RNA-seq, microarray proteomics, followed by targeted validation such as QRT-PCR, ddPCR for RNA markers, and also ELISA and multiple assays for protein markers. Those methods actually accurate, and those are highly accurate, but the issue with those methods is they are costly and time consuming. Also, they are sensitive to batch effect, and it also depends, the quality of the data also depends on the person who is doing the experimental analysis. On the other side, computational approaches actually required to narrow down the research area for wet lab scientists, and also analyze the data that are produced in experimental experiment. So let's talk about the most common computational approach, differential gene expression analysis. It is widely used, and this is one of the most popular way of identifying biomarker genes. Share. Yeah.
Okay,
This is shared over there.
We'll mark the node.
See the.
See the screen.
Okay. Can you see on Zoom?
Yeah.
me try on my phone. Yeah, it does show this thing. Should I pause for some time?
So could you see it? Good.
I can also see it.
Okay. You continue.
Yeah, thank you. Sorry for the inconvenience. So I was talking about a differential expression analysis. This is highly used to identify biomarker genes. But one of the issues with this method is it only considers a gene's perturbation across diseases and conditions. It misses all the gene-gene dependencies or contextual dependencies. There are some researchers, they use machine learning based feature selection method like a random forest. But random forest also misses individual gene effect. So based on this information, we can say that computational method either always capture only one side of the biology. To overcome this, we have introduced attention-based transformer to identify biomarker genes. When we talk about transformer, the first thing that comes into our mind is large language model. So this is one of the first work that we are trying to implement this attention-based transformer in genomics. It first came in 2014, and initially it was used to dynamically identify the words that are important in a sentence. Later, in 2017, researchers come up with a transformer version of this attention model. They use a dot product, and the core mechanism of this transformer is it considers pairwise relationship, and it answer a question that how strongly each word are connected to each other. So if we think about our genomic data, for our case it's RNA-seq data set, we can think of a sentence, and each word or token is like our genes, and the whole sentence is like our expression profile. This is how we will implement our attention-based transformer to identify biomarker genes. So based on this evidence, we hypothesize that modeling gene expression through a dual attention transformer that simultaneously capture independent molecular signals and network-dependent regulatory interaction will significantly improve the identification of biomarker genes compared to conventional method. So what is DIGAF? DIGAF stands for Dynamic Gene Attention Focus, and it integrates two models, Model A and Model B. Model A try to identify a gene's individual effect, whereas Model B try to identify gene-gene interaction effect. And also, as a deep learning method or a transformer, it can handle high dimensional data, and we all know that in genomics, the number of genes is way more higher than the number of samples available for analysis. And this method also can rank genes based on their importance. Now let's look at Model A. So as I said earlier, Model A try to identify the gene's individual or independent effect on a disease or conditions, and it try to ask the question how important a gene on its own. And we have used element-wise multiplication to implement this idea in Model A. But we all know biology is not one-sided or depends on one gene only. So in contrast, we also integrate Model B. Model B considers gene pair-wise contextual dependencies. So it tried to answer the question that which genes are important while they are in combination or while they work in a network. And to implement this model, we have used dot product. It's a transformer style attention mechanism, and it can calculate all the genes pairwise relationship. Now the question is, once we got a model A and model B, we will get two different weights for each gene. So the concern creates that how can we combine these two model into one, and to do that we have implement a novel combination metric. And the good side of this novel combination metric is it considers two sides, two real cases. The first case where both model agrees that a gene is important, or both model says that this gene is not important either for individual effect or either for gene-gene interaction effect. And in the second case, both models disagree. One model says that this gene is important, and another model says that this gene is not important. So our novel combination metric will help us to capture both cases and create a single weighted value that we can use for later uses. Once we got our combined weighted value, we will multiply this with our original input expression profile, and we will get a final weighted value of our gene expression data. And on top of that, we will use random forest to rank gene based on their importance. And now the question is why we use a machine learning model over here. Because a machine learning model works well for classification or feature selection when the number of sample is low, and it is robust compared to deep learning model if the sample is small compared to other data set. So this is the overall architecture of our method. We can see that this is the input, and once the input simultaneously go through model A and model B, both model creates their own weighted value for each gene. They are combined using our combination metric and multiplied with the original input data, and we got final weighted value. And on top of that, we have used random forest to rank gene based on their importance. Let's look at the dataset that we have used for this analysis. We have collected 259 samples from COVID-19. 90 of them are COVID patient samples, and rest of them are from healthy patients, healthy individuals. And the dataset is an RNA-seq dataset, and we also follow the typical conventional alignment procedure like quantification, training, alignment, those. And we also perform TMM-based normalization to reduce the batch effect. And one thing I want to mention that the genomes of human genomes contains around 20,000 genes. And our target is to find out the most important biomarkers that are related to COVID-19. Let's talk about the comparative method. We have introduced 4 different methods to compare with our novel method, and this includes traditional differential expression analysis and random forest-based feature selection, along with 2 modified versions of our method, where we have used support vector machine on top of the final weighted value, and another is k-nearest neighbor. In training setup, we also use multi-head attention, and we explored 2 to 8 heads, and we found that 4 heads provide us the best results. And each head trained around 239,000 parameters. Let's look at the results. So after applying our DIGAP method on the dataset, we have identified 923 genes and also ranked them out of almost 20,000 genes from the genome. And we have considered top 100 genes for our downstream analysis. This is a variable and it's flexible for different research goals. We will use these top 100 genes and their scores to validate our model as well. So we have used 2 different validation processes. One is using diagnostic or classification. So we know that our method's goal is not classifying COVID and healthy individuals, but we can get an idea how separable or the power of classification of each model. So after diagnostic results or classification, I would say, we get high accuracy 94.23, which is highest compared to other methods. Also, we get higher F1 score 96. The most important thing, we have got 100% sensitivity. That means no false negatives. And in diagnostic, we all know false sensitivity is an important feature to be considered, because if a COVID patient falsely diagnosed as he doesn't have or she doesn't have the COVID, he would have died without treatment. So 100% sensitivity is really important, and we got 100% sensitivity. We also use information theoretic method to validate our model, and this part is quite important. And we have validated both our combination metric and our models. So first of all, let me mention that our total dataset contains 439.82 units of mutual information in the dataset. So in table 1, we can see we have compared multiple combination metric, the conventional one like geometric mean, border count, harmonic mean, and it compared with our novel combination metric. So after comparing all of them, we can see that we got higher separability compared to other combination metric. Also, our combination metric can produce more mutual information compared to other combination metric. So combination metrics got outperform the conventional method. Secondly, we also compared model A, model B, and differential expression analysis, as this is the most popular one to identify biomarker. So we can see that model A and model B, they individually performs quite bad, but when we combine with our combination metric, we got highest number of mutual information, even better than differential expression analysis. So then we try to look at biological relevance of our top 100 genes. So we performed gene ontology analysis. We'll see this analysis across all chapters. This is very common analysis we do in bioinformatics. So we can see that in gene ontologies, our method comes up with higher statistical value. Biological processes like interferon response, viral defense, and cytokinin signaling. Along with that, we perform K pathway and wiki pathway, and we can see that COVID-related pathways, COVID-related pathways and common very similar disease like influenza, they came up with higher adjusted p-value compared to other compared method. So our method has also some limitations, considering that we don't have enough data. And secondly, our method only considers pair-wise relationship. It misses conditional synergy. What is conditional synergy? So in complex biological system, there could be a situation where gene A can be activated only if gene B and C co-activated. So our model can't capture this particular prospect of the biological system. So for future work, we have proposed attention-guided triple gene interaction terms, which will allow us to perform to capture the conditional synergy. And we will also look at triple gene interaction thing in my third chapter. Then we will get an overall idea that how triple gene actually works and how this will get the conditional synergy. To summarize digraph, we have introduced a dual attention transformer model that can consider both independent and dependent relationship of genes, and it can identify biomarker genes compared to traditional models. And we also proposed a novel metric that can capture both cases, agreement or disagreement. and also we saw that our method outperformed conventional methods. And the most important contribution of this work, I think, is the using large language model in genomics. I believe that this work is one of the first work that use transformer-based model to identify biomarker genes. And recently, Google DeepMind, they came up with a method and they published this paper at the end of January. They used transformer model to analyze DNA sequences to predict different prospects of biological system. But our paper has been published last year. So I believe that this is one of the first work have been done with large language model. Chapter 2. One of the critical question is how do we able to trace down downstream gene of a signaling pathways, and this is exactly what we will address in our chapter 2, and it is titled as SignalPath-Finder, identifying TOR complex downstream target genes via pseudopeak ranking and autoencoder based optimization. So again, deep learning. And this work was funded by Department of Energy, and the manuscript is under revision. We got some review from the journal. Hopefully, finger crossed, it will be published by next month. So to understand the goal of this study, let's look at some key questions. What is TOR? TOR stands for target of rapamycin, and it's a conjugate serine threonine protein kinase in eukaryotes. Second, how does it function? TOR function as a signaling sensor by sensing various environmental and internal signals such as light, nutrients, oxygen, stress. And TOR initiate a signaling cascade that can lead this type of signals to multiple downstream pathways. Some of the pathways respond immediately, some respond gradually. Next question, why TOR is important? We need to mention that TOR is a master regulator. It controls growth, autophagy, translation, hormone pathogens, and many more. But it is not a transcription factor. Instead, it's a signaling transducer that modulate gene expression profile of downstream targets indirectly, more often through phosphorylation of protein and control of translations. Fourth question: why it is so? Why does it call a complex? So TOR acts as a multi-protein complex. Rather than act alone. In many species, there are two known complexes: TOR complex 1 and TOR complex 2. However, in plants like Arabidopsis, we have only TOR complex 1 functionally characterized. Although, interestingly, we found that TOR2 homologous is present in popular Trichocarpa, but the presence of TOR complex 2 has not yet been established. And so, in this study, we will only focus on TOR complex 1 of Trichocarpa, populus Trichocarpa, and try to identify the downstream gene of TOR complex 1. It is worth mentioning that TOR complex 1 contains 3 genes: TOR1, LST8, and Raptor2 in populus Trichocarpa. But the problem is the identification of downstream gene is not straightforward. It has several issues. First, its TOR acts as non-linear, multi-step, context-dependent cascades. Second, there is no specific TOR perturbation data available for populus Trichocarpa, such as knockout or overexpression. Third, different genes response at different time. Fourth, there is simply no gold standard method available to identify downstream gene in computational area. Let me discuss briefly about how computational method falls short. Traditional approaches like differential expression analysis or co-expression analysis, they can identify immediate changes, and they mostly misses all the indirect or delayed responses. On the other hand, network inference method like Trigate, Gene3, Arachne, they assume linearity, and they also tend to remove the indirect interaction. That means we are interested in delayed response, as we know that some of the store signaling pathways response delayed. So we can't consider this network inference method as well. To overcome these challenges, we have developed a method called SignalPath-Finder. Let's look at the dataset that we have considered for this analysis. First, we have collected 849 samples of popular tritocarpa across various tissues first. But then, after manual curation and based on our research goal, we have used 628 samples across stem differentiating xylem, leaf, and xylem. And it is also, I should mention that Populus trichocarpa genome contains 34,699 genes in its genome. I also collected few known downstream genes that are already being validated in different experimental analysis. And it is 99 known downstream genes. We call it known downstream gene or KDGs. So now the concern is how do we extract meaningful signals from this noisy public data set? So the main idea over here is we try to reorganize the data in a way that it can reveal hidden signals, such as for each TOR anchor gene like TOR1, LST8, and Raptor2, we have sorted all the gene expression values, and then we divided them into equal groups. We call each group as a pseudo peak, and each group has its own sample size that are equal actually. And finally, we optimize these peaks using Grady swap optimization method for over 10,000 iterations, so that they can follow a no, I can control from here, so it doesn't work. Okay. So that it can follow near normal or near normal shape. So after the transformation, we can see in the figure that they got peak-like pattern, and after that we also reorder all the other genes based on those TOR anchor genes. We also perform some statistical analysis like Anderson-Darling statistics, then quantile error, and fraction passing to identify the best sample size for each peak. Like we can see that 40 samples in each peak. So for AD and QQ, we got the lowest one, and for fraction passing, we got the highest one. So samples 40 for each peak has been selected for stem differentiating xylem finally. Now the question is, does this peak really reveal any information for us? So we compared few of the known downstream gene, as mentioned over here, with all TOR anchor gene, and try to look at how they behave. And we can see that they showed similar kind of shape and almost peak everywhere. So based on this information, evidence, we need to recognize those peaks and quantify them as well. To do that, we have produced 4 different features for each peak. They are normality, like behavior, similar distribution, and peak weight. We also assign weight for each features, and it based on how much information they carry. Like the rare but discriminative, highly discriminative features got highest weight, and where common and less informative features get less weight. Finally, we have cumulated those features as those weight for each gene and used tissue-specific threshold value to filter important genes for us. For example, in HDX, we have a peak size of 40, and after our peak-like analysis, we got 36 features in total. And after this threshold-based analysis, we found 21,000 genes almost, which is we were able to remove almost 1/3 of the genes that are not important or noisy. So does these features really tell us something? That's why we have a look at the comparison of those features, and we can see the comparison of normal and T, normal versus similar distribution, T distribution versus similar distribution. We can see that the orange bubble that are non-downstream gene, they are clustered in a specific region, and the blue bubbles, those are non-downstream gene, or we can say noise. So based on this evidence, we hypothesize that if genes are downstream of a signaling pathway, their expression distribution will resemble the characteristic shapes of the pathway's upstream anchor components. And secondly, latent space-based representation can integrate both local and higher order gene dependencies, enabling effective ranking of genes based on their contribution to the latent space. So this is the overall pipeline or architecture of our method. Excuse me. So we already discussed about the first 4 steps, and we left with 2/3 of the gene in each tissue type. So now we need to further filter out irrelevant genes so that we can get more TOR specific gene. And to do that, we have performed cluster analysis. Cluster analysis will help us to cluster TOR-like patterns in a groups. And we also use a non-downstream gene-based selection process so that we can select the cluster that really shows natural like TOR pathway, signaling pathway. And we have actually applied k-means clustering across hundreds of seeds, and we try to get the top 10 seeds, and we try to look at their KD enrichment, their inertia value and silhouette, and try to get most important seed. We can see seed 34, 42, and 96. They showed higher spaces in radar figure. Finally, based on Lorentz and Gini curves and enrichment of non-downstream gene, we have finally decided that we will use seed 96 to cluster our genes. And in stream differentiating xylem, we found that there are 3 clusters, cluster 6, 7, and 15, that are related to TOR signaling pathway. So for method, we need some compare methods so that we can compare them whether our method is working or not. And as I mentioned earlier, that there is no gold standard method to identify downstream gene. But we can use some of the highly appreciated bioinformatics method like Spearman correlation, Gini 3, and Tigers. And for fair comparison, we have actually follow the same clustering workflow and same number of genes that we identify in our previous step. And after the analysis, we found that our method always comes up with 2 to 3 clusters, and the average enrichment of non-downstream gene is high, whereas other genes, they have some inconsistency. Once we identify important cluster, our next target is to rank genes in each cluster based on their importance. So we have used autoencoder based method, and the core idea is to learn low dimensional representations that can integrate both local and higher order dependencies. And we can see on the figure, first figure, the architecture or structure of our autoencoder method. Our original input data went through encoder layer, then it went through activation function and created the latent space. And finally, this latent space went through same activation function, decoder, and reconstructed our original output. And we calculated the differences between original and reconstructed output using MAC and R-square. And moreover, the idea of using autoencoder, we will look at how much a genes contribute to the low dimensional space or latent space. If a gene has higher contribution to the latent space, that means it's an important gene for us. And we also introduce a 2-stage hyperparameter selection method. Why we use that? Because we have almost 10 different hyperparameters. We want the best hyperparameter combination so that we can get the robust method. So we have used this stage A and stage B 2-stage model. So after getting important hyperparameter, we can rank genes, and we also compare our autoencoder based result with all other methods. And for correlation, system based correlation, geometry and trigase, we actually use their own ranking system. And we found that in top 50, we always come up with 3 to 5 genes, and in top 200, 5 to 6 genes. Whereas other method barely could manage to get a 1 gene in top 50, or 1 or 2 genes in top 200. So we can see that our method consistently get higher number of non-downstream genes. So now we will validate our method based on 2 different parameters. First of all, literature evidence and empirical analysis. In literature-based analysis, on the first figure, we can see the 3 clusters we identify in stem differentiating JM. And those yellow bubble actually novel downstream gene based on literature review, and red bubbles are non-downstream gene, the 99 downstream genes that we considered initially. And we can see that each cluster has 7 to 9 novel downstream genes in top 50 genes only. And also across all tissue types, we can see the knockout curve that the novel downstream gene showed their consistency. Then we also, in our second validation process, we have removed the non-downstream genes so that we can explain whether these non-downstream genes are the main driver of each cluster or not. But after removing non-downstream gene, we found that 91 to 95% of the gene ontology terms that are related to towards signaling pathway are retrieved. Also, we have performed empirical null testing, and empirical null testing where we have created the same cluster for thousands of times and try to find out is there any cluster that can beat our method. But the good thing is no cluster, randomly created cluster could manage to defeat us. So our selection of genes in each cluster is not just happen by randomly. Later, we also do gene ontology analysis for biological relevance, to understand their biological relevance, and we can see that cluster 6, 7, they showed off ribosome biogenesis, cell cycle growth, autophagy. Those are related to towards signaling pathways. And cluster 15 has a role on cell wall organization and cell stress and defenses. Those are all related to towards signaling pathways. So we have so many limitations. Mostly mostly there is no specific specific experimental data available for us. We need to analyze the public data. Secondly, we don't have enough known downstream gene. Third, there's no benchmark method available to compare with our method. And fourth, the annotation of popular trichocarpa is still incomplete. So we need to get the help of Arabidopsis homologous gene to perform the geo and other analysis. To summarize SignalPath-Finder, we introduce a AI-driven framework to identify genes, gene complex downstream gene. And we used pseudo-peak analysis and distributional features to capture patterns for pathway, signaling pathway. And we also introduced a two-stage model selection to find the robust and reproducible performance. And finally, we have used autoencoder-based ranking, where we can capture both local and higher order dependencies in a regulatory relationships. Let's move on to the third chapter. It is titled as Regulation of Regeneration in Arabidopsis thaliana. This work was funded by the National Science Foundation, and this work published in A-Biotech back in 2023. This is actually my first work. The goal of this study is to identify transcription factors that are responsible for regeneration in plants. Plant regeneration is a vital process for survival and propagation in nature. This remarkable ability of cells plasticity and their ability to de-differentiate and redifferentiate into new cells, such as multipotent or pluripotent cells, for example, meristem or callus, they can regenerate into root, shoot, or other organs based on the context. Let's look at a diagram to better understand the developmental plasticity of regeneration. So, on the left side, the first figure, we showed the in vitro regeneration pathway. We can see that there are some tissues like leaf, root, whole plant, shoot, hypogonal. These are differentiated cells. This differentiated cell can de-differentiate via auxin-rich callus-induced medium and can form callus. Callus shows stem cell-like properties, and it has a pluripotent state. That means callus can regenerate into root, shoot, or other organs. More specifically, callus with differentiated within root-induced medium will regenerate root. Similarly, callus differentiated in shoot-induced medium will regenerate shoot. On the right side, we showed natural references tissue like root apical meristem, shoot apical meristem. But those tissues are multipotent. Multipotent means they can convert only one, they can regenerate only one cells. Like root apical meristem can differentiate in root induced medium and can regenerate root, and similar for sap. So based on these biological details, we have collected 78 samples. We can see the samples number beside each tissue type, and we followed the typical alignment procedure like quality control, training, sequencing, quantification. I also collected 145 pluripotent genes based on literature evidences, and we found 78 transcription factor, the rest are non-transcription factor. But transcription factor is important for our case because we are trying to find out transcription factors that are responsible for regeneration in plants. So we have used a method called TF cluster. TF cluster can create subnetworks or clusters, and it uses two-stage process. On its first stage, it uses shared co-expression connectivity matrix, where this matrix builds using this matrix calculates the Spearman-based co-expression between transcription factor and their target genes. We can call target genes co-expressed gene, and then count the shared co-expressed gene for each TF and make a new matrix called shared co-expression connectivity matrix. In its second stage, we decompose this ACCM matrix using triple link algorithm, where we started with the highest shared co-expression gene. We started with TF pair that has the highest shared co-expressed gene, and we iteratively added TFs until no TFs left behind. Let's look at the diagram of this method to better understand what I was trying to tell. This is our gene expression data. We have performed Spearman analysis. We can use other, but Spearman can capture the non-linear relationship. And we found this co-expression matrix. And based on this co-expression matrix, we created ACCM, or shared co-expression connectivity matrix, that are between all the TFs and their shared number of co-expressed genes. So we got a network like that. On top of that, we have applied triple link gene and triple link algorithm. Triple link algorithm helped us to separate each subnetworks of important, and finally we got subnetwork that are highly co-regulated or have a similar regulator role in biological complex system. Then we have performed PCA-based analysis to reduce and focus on important and robust subnetworks only. And to do that, we have compared the gene expression of each subnetwork along with the eigenvalue trend. If our hypothesis is if the eigenvalue trend and gene expression followed closely each other, then those particular subnetwork highly co-regulated and they link to same biological system. Based on this analysis, we have finally collected 9 collaborative subnetworks. And in the table, we can see that each subnetwork has multiple pluripotent genes. And I should mention that we didn't use the pluripotent genes to do the analysis. So it's totally unsupervised. On the figure, we can see different subnetworks. The bigger circles are transcription factor, and the white bubbles in between them and are the co-expressed gene. We can see that subnetwork 1, subnetwork 12, and 17 has the highest number of common co-expressed gene in between them. So we will forward with those 3 subnetworks only. And if we look at the tables, we got the highest number of pluripotent genes for these 3 subnetworks also. Then we have performed motif enrichment analysis. Motif enrichment analysis would help us to find the overrepresented TF binding sites in their co-expressed gene. And to do this analysis, we have used SysBP database to create the position weight matrices, and then we have applied a motif locator to scan the motifs, and finally we have performed hypergeometric test to get an idea of each gene get an idea of each transcription factor, are they significant for the motif enrichment or not. Then on top of the motif enrichment analysis, we have performed this triple gene mutual information analysis. Triple gene mutual information, this help us to rank a gene by identifying the relationship, regulatory relationship between transcription factor and their pathway gene. Pathway means, for our case, we are talking about regeneration related pathways. And we have calculated mutual interaction measures, or MIM, to assess this regulation. And finally, based on a particular transcription factor's frequency that passed these particular mutual interaction measures, or MIM, we will rank them. Let's look at the results of motif enrichment analysis. We can see that for subnetwork 1, the genes are statistically significant, but for other subnetwork, the genes are not statistically significant when we did our motif enrichment analysis. So we will focus on subnetwork 1 only from now on, and we have applied TGMI, or triple gene interaction analysis, and we try to rank all the genes in that subnetwork, in subnetwork 1, and we can see that on top 30 genes, almost 13 of them are literaturely are verified by the literature evidence, and we will look at those genes pattern. Sorry. Before we look at the pattern, gene expression pattern, let's look at the gene ontology analysis again. And we, as I mentioned earlier, that regeneration regeneration process has some related biological process like somatic embryogenesis and regeneration. And if we look at the gene ontology analysis of subnetwork 1, we can see that both regeneration and somatic embryogenesis got enriched. But in subnetwork 2, only regeneration got enriched, but subnetwork 17 didn't show any enrichment for regeneration or somatic embryogenesis. Let's look at the pattern of subnetwork 1 gene. In figure A, we look at the 3 already known pluripotent genes like LEC2, EGA37, and WOX9A. And in figure B, we have showed novel candidates, TF candidates, 10 of them. And if we look at them, then they showed similar peak in similar tissue types. But the rest of the genes, almost 24 of them, they showed some arbitrary patterns. To summarize this work, first of all, we integrated public RNA-seq data that are related to regeneration, and it helps us to capture the transcriptional landscape. Second, we have used TF cluster to identify collaborative subnetwork. Third, we have used TGMI and motif analysis to narrow down our focus. And finally, we have identified 3 known regulators and 3 most important novel candidate transcription factor in regeneration. So this is the end of my 3 chapters. Along with these 3 chapters, that I also contributed in developing Sapphire, a web tool to serve SARS-CoV-19 RNA-seq database. This is the same project like my first chapter. And I also worked on two different methods, Integra gRNA and TG-PRET. Those are already published back in 2023 and 2025. And I had the chance to work in lab facility as well, where we try to experiment low and high nitrogen. I also had the chance to guide two graduate research helper. And currently we are working with TOR1 and TOR2 transgenic line in Populus tremula, and with collaboration with Professor Gary. Acknowledgement. So first I would like to thank my advisory committee for being so kind to serve as my advisory committee, as well as their suggestion, their feedback helped me to shape myself and my research as well. I would like to mention specifically Professor Hui Jiang for his immense help during my job hunting and his direction for my research. And I would like to also thank Professor Shah. I have took two of her courses, time series analysis and predictive analysis, predictive modeling. And I learned a lot of them, and you can see I can also try to apply some of the context over here. And I would also like to thank Professor Weo Zhou. He's not in person today with us, but he is online. He helped me a lot to shape my final chapters during my proposal defense. And I also would like to thank all my classmates and colleagues for different topics, sharing knowledges, experiences, for making my journey a little bit easy in a place like this, snowy. And I would also like to thank all the funding sources for making my research possible. And most importantly, I would like to thank my family, more specifically my parents. My dad used to tell me, how long will you be a student? Maybe today I can tell him that I would be no longer a student very soon. And thanks to my mom, he always supported me. Especially I had the chance to get my higher education because of her. Otherwise, I would have ended up in a government job back in Bangladesh, which I really hate. And I would also like to thank my brother and sisters and their lifelong support. That really means a lot to me. And I would also like to thank one professor, Professor Dr. Jahidul Islam from my undergrad university. I started my research with him. He also helped me to secure a PhD under Professor Dr. Hairong Wei. I really appreciate that. And my teacher, Shadid Al-Nuri. And I want to mention some of my friends that present today. Rejawai, Rapsan, Arjun, and then Tanvir Bhai and Fariyabu. They helped me to arrange today's session. They did all the arrangement for the food. I just try to focus on my work and presentations, thanks to them. And I would also like to thank Novin Bhai and Ontu Bhai. They helped me mentally and supported throughout this PhD journey. And last but not the least, my advisor, Professor Dr. Hairong Wei. When I first joined in his lab back in fall 2022, I was just I just passed my undergrad, and I was going through this pandemic situation, everything locked down. And I got offer from him, and when I first joined him, there was so much uncertainty because I didn't came from biological sites. So I have zero knowledge about biology. So then he started to teach me biology for 20 minutes every morning, which helps me to understand biology. And now I can say I can read any biological paper. And I also learn how to make a method for a problem from scratch using statistical method and machine learning, deep learning. Also, I would like to thanks for his financial support throughout this PhD journey. And moreover, I show him as my guardian over here, not like not colleagues or boss. He treat me like his son, and thanks for that. He, yeah, thanks to him overall. And the code and the data that we have used for analysis will be available in this website. You can scan, and I'm happy to take any questions. Thank you.
Okay, so let's do this way. Non-committee might ask a question first. Committee will ask a question later. So you guys, any question?
Yeah, I have some question. Actually, I'm pursuing. I'm a biology guy, so lately I'm investigating some interesting things in bioinformatics, and I have some questions. Like in your first chapter, you use before I'm going there. I must say it's an incredible presentation and work,
you.
Dr. Hairong Wei, and appreciate sharing this huge knowledge with us. And I saw in your first chapter you use random forest to rank the genes, and in the second chapter you use autoencoder ranking the gene. My question is, what are the difference between these two systems, and why do you use these two systems? Can you tell us?
Thank you for this. Is quite a good question, I would say. For my first chapter, I have used a data set that includes both control and case. Case, COVID, control, healthy individuals. So it's a kind of supervised method. So we can use random forest to select the features and rank them. But for my third chapter, we don't have any samples that are control and case. We have like a bunch of data set collected from public resources. So we don't know which one is overexpressed, knocked out or not. So that problem is fully unsupervised. Autoencoder with reconstruction doesn't need any control case. It can work unsupervised or without any direction. So that's why I use two different methods in two different chapters. Thank you. That's a very good
I have one more
observation.
question. Actually, I'm
Yeah.
focusing on regeneration after the transient-free genome editing. So you said in your last chapter that the three transcription factor named AIL1, Y5, and PEI1. So this regeneration is from callus to shoot, or I mean, from where? I mean, yeah.
So these three genes
Yeah.
that you mentioned, AIL3, YIP5, and PEI1,
Yeah.
that are showed in figure 2. So figure B, sorry.
Yeah.
Showed in figure B. And if we look at the lesion, we can see they showed higher peaks, same as known pluripotent genes. Based on this evidence, we have selected these three as the highly promising transcription factor. We are not saying that these three only, we have 7 others as well, but based on the pattern, we are some kind of hypothesis that these three could be the most important one. Am I?
No, actually, I want to use them, so that's why I'm asking. Yeah,
That's our job to narrow down the research area for experimental guys, scientists.
Thank you, it's helpful.
You're welcome. Yep.
Thank you for your wonderful presentation. Actually, I am from different background, material science and engineering background. In the era of the artificial intelligence the incorporation of statistical analysis, artificial intelligence to solve the real problem is promising. So while you are presenting your chapter 1 and chapter 2, I am thinking about my areas as well, and like am I able to incorporate those methods to solve the problem of material science and also problem of the biomedical engineering as well. Like if I ask particularly, like is those model can be applicable to know the correlate the factors, particularly in if I change the pH and material and temperature, and how those are related for the changing main biocompatibility of the vascular stent. So am I able to like know the interrelation of those in particularly in cardiovascular stent applications if I change different parameters?
To answer this question, even this method that we used for genomics actually used for natural language, large language model to learn the context of language or sentences. So the major important cases that need to be done to apply a machine learning or deep learning model on different field depends on how you recognize your features. Like in your case, you said that you have used, you want to use pH, temperature, those. For our case, our features is gene. So gene A, gene B, each gene has its own role, like temperature and pH has. So the most important part of applying deep learning or machine learning is to know how your data set can be fit to a particular model. So if you can analyze the data in a way that it makes sense, like your features are temperature, pH, or other biocompatibility, and you have multiple samples, then you can obviously use different machine learning and deep learning methods. So it's all about knowing your features, the attributions of your data set or samples.
Thank you so much, and good luck for your next endeavor in your postdoc journey.
Sure, thank you.
Okay, no question? No? Okay, so you guys might pick up something in the back, so you know, we will discuss.
My way to like last day, actually,
Help yourself.
for you, you need to ask everyone to leave the Zoom as well, and only
Pick up.
Professor will know Zoom.
Once we finish, we can get some.